So meetings being live streamed. Go ahead. So we'd like to thank a lot Dr. Alessandra Pontillo for accepting our invitation to present her work here in the Immuno webinar series. So she's an associate professor of the Institute of Biomedical Sciences at University of São Paulo. Dr. Alessandra Pontillo got her degree at University of Trieste in Italy. Then she moved to Brazil after completing her postdoctoral training, and she's being a professor in our institute, and it's a pleasure to have her here to share some data with us. Thank you very much, Dr. Alessandra, and you're on.
Okay. Thank you, Diego and Gustavo, for the opportunity. I think it is a good opportunity for all of us here of the department to may share our research just to has example for our cooperation project in future research. So today I hope to be short, but I just present a part of our research that is main focus on genetics, but of inflammasome. And our main focus in the lab is have two main parts. That is a part that was clinical, that was the diagnosis and characterization of autoinflammatory diseases, and in particular rare diseases of inflammasome, that are the CAPS, that was of the NLRP3 mutation, and the other monogenic disorder. And we have a more basic line of research that it was what we mainly do in the lab. That was projects that are started from by a genetic point of view, so with the association studies, and then that are some of them going to the in vitro part to characterize the effect of the genetic variants on the inflammasome activation. And so the two main questions that we are trying to ask are if the same genes that are mutated in the rare disease, in the inflammasomopathies, may be responsible also for the contribution to the pathogenesis of multifactorial diseases that are more common in the general population. And studying these multifactorial diseases and also the rare diseases, we also have a second question that is maybe the most intriguing for us, that was what about the inflammasome activation in different cell types. So here today I just try to be short, but I would like to share our results about these first two topics. That was the dysregulation of the NLRP3 inflammasome and in myeloid cells and lymphocytes, and what about these quite new described pathways of inflammasome activation, that was the NLRP1 or CARD8 pathways in lymphocytes. And the third, I think that I don't have the time to discuss a lot about, but it is just what we are planning for the next future. That is what is, in our opinion, the function of CARD8 in the regulation of the inflammasome. So, okay, so first I would like just to resume briefly, okay, I think that inflammasome is it is been known from a lot of you. I think the majority of you, it is a very known complex, okay, so we have different type of intracellular receptor. Maybe the most famous, it is the NLRP3 receptor, but we have also the NLRP1 and the CARD8 that here it is not described, okay? And when these intracellular receptors are activated by several types of cell stress, we have the organization and the mounting of the inflammasome complex, the activation of the enzyme that was the effector enzyme, the what caspase 1, and the cleavage of several substrates and IL-1 beta, IL-18, and Gas-dermin D. And what happens when we have rare mutation in some of the receptors? We have that the inflammasome complex is hyperactivated, and we have an excess of IL-1 beta, IL-18, and so we have a systemic inflammation that, curiously, it is not the same depending on the receptor that is mutated. So this is the first intriguing point, that we have, depending on the receptor that is mutated, we have a clinical heterogeneity between the autoinflammatory diseases, and probably because these receptors are differently expressed in different compartments of our body. So depending on the cells and on the tissue, we have different effect of the mutation in the patients. So this is the first point. Okay. And okay, so when we are trying to characterize what happened in these patients with the rare mutation. Here I just show you the example of the NLRP3 mutated patients. These patients that have this CAPS syndrome, they have a gain of function mutation in the NLRP3. So we have this excess of IL-1 beta and IL-18 that is characteristic of the patient. But when we are looking at in vitro behavior of the cells of this patient, we just see some intriguing behavior that is very typical of these patients. The patient, here are monocytes that are isolated from the patients, okay, and that are in red here, the CAPS monocytes, and here in blue we have the healthy donors as a control. And you see that in healthy donor, we have that when you treated the monocyte with the lipopolysaccharide, okay, you have just an increasing in IL-1 beta production. But when you have the ATP that it was adding to the assay, you have a great increase of IL-1 beta. And this is a very, very characteristic behavior of the NLRP3 activation, okay? And normally this production of IL-1 beta, it is increased in more than fold respect to the LPS. So it is a very, very big increase of IL-1 beta. However, in the CAPS patients, in the monocytes of CAPS patients, you see that really the IL-1 beta is really increased also in the LPS condition, but we have no increasing when you are adding the ATP. So this is the profile of a dysregulation of the NLRP3 in monogenic disease that was the CAPS patients. Okay. Okay, so it is very interesting that when we are trying to characterize a Brazilian patient of CAPS, okay, and we are the only, at the moment, the only group that made this type of characterization, we have that really the NLRP3 mutated patients have this lower ratio of IL-1 beta between LPS and ATP and LPS condition, okay, so lower than 2 times, okay, but this ratio, it is the same when we have patients that are negative to the genetic screening. So this is also the second point that is intriguing, and it is very known, okay? It is not just for the Brazilian patient. We have the same, quite 50% of CAPS patients that are negative for the NLRP3 mutation. So the point is that we have this characteristic profile of dysregulation of the NLRP3 inflammasome, that it is the same when we have the mutation or not. So this is the point. What is the contribution of the host genetics and what is the contribution of the environmental? Okay. And starting from this observation, okay, quite 15 years ago, we are starting wondering if this dysregulation of the NLRP3 inflammasome could be not just associated with these monogenic diseases that were the CAPS, but also with the multifactorial diseases, because we know that the common diseases such as neurologic disorder or metabolic disease, cardiovascular disease that are very common in the general population, okay, they are also associated to inflammation, not frequently with the high level of IL-1 beta or IL-18. And in our genetic study, we have demonstrated a lot of association with common variants in NLRP3 gene and this type of diseases as a contributing factor in the pathogenesis. So the point is that when we are looking at the localization of the mutation, you see that here below we have the mutation localization. So these are the variants that are founded in rare patients, okay? But we have also some variants that are very commonly present in the general population, such as this variant that was localized in the UTR region of the that was present in quite 30% of the population. So it is not very low frequency in the general population. And what we know is that this, in particular, this variant, we are working with other variants, okay, but these variants, it was associated to several types of disorder, infection diseases, such as HIV, but also not infection diseases, such as type 1 diabetes, celiac disease, and so on. So the point is that, okay, but what happened in the cells, okay, of the host when the host is carrying some of these common variants in the NLRP3 genes? Because the most of our understanding about the NLRP3 inflammasome, it is based on assays in monocyte macrophages, okay, or in cell lines in human cells, okay, and but really the genetic is the same in all our cells. So the point it is what happens in our cell when we are carrying this type of variants. Okay, so here I just put an example just to know this variant. It is really associated with different NLRP3 inflammasome activation. This is the so-called genotype guided assay. So we have the same assays of when you are activating the monocytes with LPS and ATP, and you are measure the IL-1 beta production, and we are just comparing the donors that carry or not carry the variants of these variants of the UTR in the NLRP3 genes. And you see that really the polymorphic allele, that is the G allele, it is associated with higher level of IL-1 beta. So this variant, it is associated to several diseases as it is associated to higher level of NLRP3 inflammasome activation and IL-1 beta release. Okay. And okay, so the point here, the question here are two. The first question is that, okay, so we have this genetic effect that was the gain of function variant on the NLRP3, and we have the question of the environment, the medium in which the cell it is exposed. And we are just trying to look at the environment of different conditions. For example, this is the first graph here. It is just the same of the CAPS patients, okay? Just to remember that the CAPS patients have this dysregulation of the response to the LPS ATP challenge. And when you are seeing the monocytes or dendritic cells derived from monocytes of HIV patients, you see that it was in pinky, or the macrophages derived from monocyte from people with active tuberculosis, okay? You observe that a very, very common behavior, okay, that was the incapacity of this higher response to the ATP on the LPS challenge, both in the HIV and tuberculosis patients, okay, and also in these different type of cells, but there are also all myeloid cells, okay. And it was intriguing this, and when we are, more recently look at the cells from severe COVID patients, okay, in that case, we are looking at the neutrophils. Here, we observe quite the same behavior. So the point is, when you are in a very inflammatory environment, you have this sort of dysregulation, okay, so a lower response. Normally, it is a higher constitutive activation of the NLRP3 pathways, but a lower response to another type of challenge. Okay, so this is the first point. However, we are trying to better understand this question of the inflammation. It is it is an inhibitory factor for all the pathways that activate the inflammasome, or it is specific for the NLRP3, as it was demonstrated for the monogenic disease. Okay, and so we have these data that are from PhD students of the lab, okay, Edioni, that it was try to understand if in dendritic cell from HIV patients, and here we use HIV patient just as a model of chronic inflammation, not just for the infection itself, because all the patients are treated with antiviral drugs, okay, but they are constitutively inflamed. Okay, so the point here is that when we are treated dendritic cell from HIV patients, you see that we have quite a similar response to flagellin. That was one of the way to activate the NRSC4 pathways, okay, in HIV, that was the black bars here, and in the healthy donor. So apparently, in HIV cells, we don't have an inhibition of the NLRC4 pathways. And here also we have tried to down-express the NLRC4, okay, with shRNA or NLRP3, and you see that really we have a complete functionality of the NRSC4 pathways compared to the NRSP3 pathways in these patients. So the point is that really here the defect it is specifically to the NLRP3. Okay. Okay, so the second point is that now I just show some data from Vinicius, that he was the postdoc in the lab. And okay, so the first point is that what about this question of the neutrophils and the NLRP3? So the NLRP3 have this common behavior in monocytes and monocyte-derived dendritic cells. But what about the other types of cells? So we are just looking at the neutrophils, okay? And really in neutrophils of COVID patient, we have an increase of constitutively activated NLRP3 in severe COVID patients. And this activation of the inflammasome that was observed here by the activation of caspase-1 in the neutrophils and by ASC-SPECS aggregates here, it is inhibited in the presence of the inhibitor of the NLRP3. So really, NLRP3 is present, okay, in the neutrophils and could be dysregulated in these cases of patients. Okay. What about the HIV? That, as I mentioned, it is one of our models of chronic inflammation. Here we have just ongoing experiment, but neutrophils from HIV patients also have this increased activation, basal increased activation of the NLRP3 that was observed by these ASC-SPECS here, okay, in the HIV neutrophils that appears to be activated and just with nets and dysgranulating cells compared to the healthy donor cells. And we are just curious about what happens in the activation, because really it is, we don't know if the neutrophils of HIV patients behave just as the monocytes and monocyte-derived cells with this inhibition in the activation or not, because this is the point, if we have different types of regulation of the NLRP3 pathways depending on the cells. Okay, and Vinicius, it is just looking at more specifically to the activation of the inflammasome in the neutrophils. Okay, and it is he's just describing that we have a difference in the activation of the NLRP3 inflammasome in the neutrophils compared to the monocytes and macrophages, okay? And in particular, one of the pathways of activation of the NLRP3 in neutrophils that was mediated by the Bruton tyrosine kinases. Okay, it is acting in opposite way in monocytes and in neutrophils. So normally we just know that these kinases is important to amplify the NLRP3 activation, okay, normally through the so-called alternative pathways that is mediated by dectins or these receptors that are PRR receptor on the cell membrane. And through this phosphorylation, normally this phosphorylation signal, it is activating, no, it is amplifying, it is not necessary for activation, but for amplifying the NLRP3 inflammasome activation. However, in neutrophils, he observed an opposite role of this kinases that apparently it is inhibiting, so it is just making a break on the NLRP3 activation. So it is just for you to know that really when we are talking about the NLRP3 pathways, we have different mechanism of activation depending on the cells and different behavior depending on the condition of the cells, if it is in a healthy donor or if it is in the patients. Okay, so then we are just looking at the lymphoid compartment, because in the recent year, some people have shown that the inflammasome, it is not just present in the myeloid cells, but also in the lymphocytes. And we are using again these HIV models just to better understand first these pathways of NLRP3 activation in B lymphocyte. So we are isolated the CD19 cells from the patients, and we are just looking at what is happens. And okay, so in the basal state, the B cells are activated. Okay, we know that HIV condition, it is a pro-inflammatory condition, so we normally have all the cells that have a pro-inflammatory profile. And you see that we have in the basal condition just a little increase of IL-1 beta production and caspase-1 activation without cell death. Okay, so and what happened when we look at the activated condition? So here we used the beta-glucan as a activation of the dectin-1 receptor, so that this was one of the alternative pathways for the NLRP3 activation, okay, that it is also involving the Bruton tyrosine kinase here. And we see that activating the B cells of the HIV patient, you see that you have an increased response for the IL-1 beta production, okay, and the inflammasome in general. I just put this for you to see. And this type of activation, okay, it was inhibited by the addition of our assay of the MCC, that was the specific inhibitor of the NLRP3, okay, and this other that was the CGI, that was the inhibitor of the Bruton tyrosine kinase. So here we just confirm that this pathways is working on B cells, that it was just published by another group, okay, but we also see this regulation, in that case, a upregulation of the NLRP3 pathways in B cells from the HIV patients. Okay, so this is the first result that we have as an opposite result compared to the myeloid behavior in HIV patients. When you see also, okay, in the first group that published about the NLRP3 pathways in the B cells have linked also this activation of the NLRP3 inflammasome to the increasing release of IgM. And so we are just looking also IgM production. This is, and just to show that also in HIV patients, you have an increase of IgM production that was in parallel with the IL-1 beta production, and that was dependent to the Bruton tyrosine kinases here, more than on the MCC. Okay, and here we are just wondering if what about the genetics? We know that these common variants on the NLRP3 genes, that was the three lines UTR variants, it is associated to the pathogenesis of HIV. So we are wondering if it affects also the B cells activation of the NLRP3 inflammasome, and you see that the IL-1 beta production by the CD19 cells, it also was affected by the genetics of the NLRP3. Okay. So we are just moving to another type of lymphocyte, the CD4 lymphocytes. Okay, so again, we have at the time, we have just this paper of the group of Kemper that it was showing that in healthy donor, we have an NLRP3 activation of inflammasome through the TCR complex stimulation. Okay, and that this stimulation of the NLRP3, it is leading to the IL-1 beta production and of amplify the production of interferon gamma in a not very clear way. Okay, so we are just looking at our HIV CD4 T cells, and you see that in HIV, you have, we challenge the CD4 positive T cells with the anti-CD28 and CD3. And you see that an increase of caspase-1 cleavage, okay, in both healthy donor as expected, and it was increasing in HIV patients. So once again, here we have opposite behavior of HIV cells, lymphocytes compared to the myeloid cells. So an increasing activation of the NLRP3 pathways. When we are looking at the IL-1 beta, also the CD4 from HIV patient are hyperactivated. Okay, so it was an increased release of IL-1 beta. And when we are looking again to our common variants here in the NLRP3 UTR, you see that also in that case, this trait of IL-1 beta production in this condition is affected by the genetics of the donor. Okay, so it is a gain of function. Okay, in this work of Kemper, they are not very interesting about the pyroptosis at that point, but we are just looking at if this activation of the CD4, could be also related to the increase of pyroptosis, and it is in the CD4 here, as we see by the LDH release. Okay, and you see that the NLRP3, it is really the actor of this response, because we have these specs that was here. We have the specs of the NLRP3 as we see of the HUS, but I just show you this NLRP3 to show that really this activation through the TCR complex, it is mediated by the NLRP3. And here it is, again, when you are inhibiting the NLRP3 in these cells with the MCC, you see that the IL-1 beta, it is decreasing. So really it is, this pathway, it is activating the NLRP3, and the NLRP3 in lymphocyte, it is hyperactivated in HIV patients. Okay. So again, and we are just now looking at the more recent results that are these from the PhD of Mariella, and she is looking at CD8 T cells. Why we are looking at CD8 T cells in HIV patients? Okay, it is because what we know about the NLRP3 in CD8, it is very, very poor in the literature. The group of Kemper was not able to finally detect really an activation of the NLRP3 in CD8 T cells, and really we have also no success with the activation through the anti-CD28 and anti-CD3 in the healthy donor CD8 T cells, so no activation one, and a very poor activation of specs. So really the group of Kemper showed that through these pathways, really it is apparently the CD8, it is not responding. However, when we are looking at HIV patients, we see an effect. Okay, so again, this chronic condition of the HIV patients, it is making something in the CD8 cells and increasing the response of the NLRP3 inflammasome in CD8 cells. So here it's just to see the activation of caspase 1 in red bars, that was the HIV patients, and the increasing of ASC specs. And okay, the point is, okay, for sure CD8 cells don't make IL-1 beta. So we are just trying to see another read for our experiment. So thinking about the result of the Kemper group, we are just looking at interferon gamma, okay, that we know that it was increasing normally in the HIV patients, that it was the same here. Okay, but okay, so how we try to connect this question of the interferon gamma with the inflammasome. So we are just including in our assay the inhibitor of caspase 1 here. Okay, and you see that the interferon gamma production, it is reduced, and the same when we are using the MCC inhibitor that was inhibiting the NLRP3. Okay, and interesting, okay, when we are looking at the genetic of the host, also we see that not the 3 lines UTR variants, but these other variants of the NLRP3 that was a missense variant, it is also affecting the production of the interferon gamma. So again, we have a condition, the HIV condition, the environmental of the cells, and the genetic of the host that can show that the NLRP3 is making something in the CD8 cells. Okay, so what we are also looking at pyroptosis, okay, for the CD8, and really here we see that in the healthy donor we have quite no response in terms of PI uptake. Okay, that one of the method that we are just accompany the pore formation in that cells, but you see that in a red curve that in the CD8 from HIV patients we have an significant increase of pore formation. Okay. However, this pore formation, it is not associated to a LDH release. Okay, so the question is, okay, so a pore it is forming there, but we don't know if the pyroptosis is really happen, and we are trying to stimulate it again, our system, just to know what happens, and we added in our assay the ATP that was one of the normally activators of the NLRP3 pathways. And it is intriguing because we have observed a very interesting increment of the pore formation, both in the healthy donor, that was the gray lines here, and in the HIV. Okay, so the proportion of the pores, it is increasing, and in this condition, we also see an LDH release. So really, the TCR stimulation itself, it is maybe not sufficient to activate the pyroptosis, but when a dump such as the ATP, it is released in the medium, you can observe this effect of the LDH release. And again, when we are looking at the genotype of the NLRP3 variants in this experiment, we also see that here it cuts the variant, but it was the same, the missense variant in NLRP3 genes. Also, you have an increment of PI uptake. So it really, we can observe this situation of the host genetics that affect the activation of the NLRP3 pathways in CD8 cells, and also the question of the medium of the cells. Okay. Okay, but in the recent years, we have also another surprise from the literature, that it was that another pathways of the inflammasome, that it was the NLRP1 or CARD8 pathways for the inflammasome activation, was discovered not just in the epithelium or in the monocyte and macrophage, but also in the CD3 lymphocytes. Okay, and the authors that describe these pathways in CD3 lymphocytes, they are try to better understand what is happened when you are challenge your lymphocytes with this chemical that was the Valboropro, that is an inhibitor of this dipeptidase 9, that was the natural inhibitor of these two receptors. So what happens is that then when you treated your cells with this Valboropro, you have the activation of the NLP1 or CARD8 receptors and the activation of the inflammasome. In the lymphocytes, they are showing that this type of activation is inducing pyroptosis. Okay, so the question that we have is that what happened, okay, when you are working with this chemical in CD8 cells from HIV patients. Okay, so because again, we are wondering if in HIV patients just the NLRP3 it is a strangely behavior, or it is a common behavior or other pathways for inflammasome activation. And you see there that in HIV patients, okay, you have a little reduction of the caspase 1, okay, in the activated cells here, okay, that was this in the bar graph here, okay, and the question here is that we don't know if really we have a reduction of Valboropro response here in HIV patients, okay? However, apparently we don't have an increase of response, nor a very big lowering of the response, okay? And when you see the aspects, you see also that the Valboropro is inducing quite the same behavior. So you have in CD8 cells an increase of ASC specs in the Valboro treated cells, and in HIV you also have an increasing of ASC specs, but apparently you have a reduction of compared to the healthy donors cells. Okay, so okay, it was intriguing for us, but then we are just thinking that really the first group that described this pathways in lymphocyte also showed that when you are activating your T cells, you have a less response to the Valboropro stimulation. So the activated T cells are resistant to the Valboropro stimulation. And as we know that the lymphocyte from HIV patients are activated, constitutively activated, okay, we can think that this reduction in the response of the CD8 cells may be a consequence of this basal activated state of the cells. Okay, and sorry. Okay, so the question here is that, okay, what is the receptor that is mainly responsible for this Valboro-Pro response in CD8 T cells, because in the previous literature, the author showed that it was the card8, okay? However, we are thinking that it was the NLRP1. Why we are thinking about the NLRP1? Because the first question, we ask specs formation, and the card8, it is not activating the inflammasome through the recruitment of ASC. So this is the first point. And then, you see the oligomerization of the NLRP1 in CD8 here. Okay, so again, you see the oligomerization of ASC, you see the oligomerization of NLRP1, okay? And then again, sorry, the genetics came in our help, because when you are try to see the effect of Valboro-Pro in CD8 T cells, you see the increasing of PI uptake, okay? It was just demonstrated that this pathway is inducing pyroptosis, okay, in lymphocytes. And when we are looking at the genotype of some variants in NLRP1 gene or in card8, okay, receptor, you see that just the variant in the NLRP1 gene are affecting the response of CD8 T cells to Valboro-Pro. So again, it is maybe an approach that is not very conventional, okay, but we are just insisting that for us in that model, the NLRP1 is the major sensor of the Valboro-Pro inhibition in CD8 T cells. And okay, so this is sorry. So and briefly now I just show you what happens in a genetic association, and we see that NLRP1 variants are associated, genetically associated to several diseases. And so the question is that we are now have a new way to interpret these results of association, because when you associated the variant in NLRP1 with asthma or multiple sclerosis, or you are thinking about how this variant it is affecting the keratinocytes, the epithelium, or the monocytes normally. But if you know that this variant have a functional effect in lymphocytes, you can rethinking about the pathogenesis of the diseases and how the NLRP1 variants may affect the pathogenesis of disease through the affecting the lymphocytes and not just the monocytes or the epithelial cells. So this is the question that is intriguing when we are looking at these new results, okay? And similarly, we are just looking at our association study in some B-cell mediated diseases, okay? And here we are just our previous results about the juvenile lupus and asthma. We have made this study a lot of years ago, okay? However, now we are just reanalyzed some points here that are the production of antibodies, okay? And you see that this variant that was in the card8 and these other that are variants in the NLRP1 apparently affected the antibodies production in these diseases. So we are just looking obviously how the NLRP1 and card8 pathways are playing some role in the B cells. Okay, So here there are very recent results. So we have just looking at the Valboro-Pro response of the healthy donor B cells, and you see that really this pathway is working also in B cells, as you see here for the activation of caspase 1. And it is not different depending on the naive or memory circulating B cells, so it is the same response. You see that these pathways activate the ASC specs, so again, it is also apparently activated the NLRP1 pathways, and you see that differently to the beta-glucan that is working through the NLRP3, these pathways in B cells does not activate the IL-1 beta production, okay, but it is activating the pore formation, as it was see here for the uptake of the PI. And the PI uptake and LADH release, showing that this stimulation is also inducing pyroptosis in that cells, even if the quantity of LDH release, it is not just very great respect to the T cells. Okay, so it is apparently they are more resistant to pyroptosis in these same conditions. But this pathway is depending on the caspases, and these are two inhibitors of the pore formation that was made by the Gasdermin D. So really exist these pathways also in B cells, and we know that also in that case, these variants of the NLRP1 are affecting the pore formation, okay, in B cells, and not the CARD8. So again, we are thinking that also in B cells, even if the two receptors are present, our data are favorable to an NLRP1 activation more than a CARD8 activation in these cells. Okay. Okay, but I just finished now with a great question that, okay, so we are just showing that the NLRP1, it is activating in the T cells, in the B cells, but what is the function of this sensor in that cells? Because the recent literature showing that these receptors are sensor for viral proteases, okay? And so the question is, exists some virus for the B cells or T cells that can activate this sensor naturally, okay, not in an in vitro model, okay? And we just thinking about the Epstein-Barr virus for the B cells, and we just see that exist really two protein of the ABV virus that can cleave the human NLRP1 protease target, okay? So really, it makes sense that NLRP1, it is expressed and doing something in B cells because maybe it was just a host protection against this B cell specific virus. For the CD8 T cells, we don't know. So really, we are just trying to understand if maybe it is not a sensor for viral proteases in CD8 T cells, but maybe it is could be activated by some stress, such as redox stress or other type of cell stress that are normally has been associated to the NLRP1 or CARD8 activation. Okay, so concluding, I think that we have just a lot of stories that are driving parallel in our lab, but in these 10 years, we have just seen that really the NLRP3 pathways for inflammasome activation it is differently regulated depending on the cell types, okay? And we know that we have several steps in which the NLRP3 could be regulated. So the priming step, but also the post-traducional steps and some step of ubiquitination, deubiquitination, phosphorylation that can be important for the activation or the entity of the activation of the NLRP3 in different cell types, okay? And it is also important to understand that each of these proteins that can be regulated, the NLRP3 in different cells, also can be affected by genetic variants. So the point is that the next step for us, it is not to just see the genetic variants in the inflammasome census, but in the genes that are upstream the NLRP3, just to understand how, if they can modulate the NLRP3 response in different cell types. And the second point, it is that we are insisting that the NLRP1, it is important in the response of Valboro prostimulation in the T cells, okay? And it is, okay, the literature showed that it was a CAR-A activation in healthy donor T cells. However, the two receptors are present and equally expressed in the T cells and B cells. So once you are just stimulated with Valboro Pro, why just one of the two receptors may be activated and the other no? Not so. We are thinking that probably the two could be activated, but in some condition, the NLRP1 could be the receptor that have the majority effect, such as in of HIV patients. Okay, and thinking about these two pathways, the NLRP3 pathways and the NLRP1 pathways, the third point that was the next steps of the lab, that was how CARD8 do within these two receptors, okay? Because, okay, 20 years ago, people tell us that the CARD8 was an inhibitor of the NLRP3, and that we have some variant that it is a nonsense variant, so that skip the CARD8 from the cells, and it was activating the NLRP3. Okay, and recently other authors showed that the CARD8 isoform, some of isoform, of splicing different isoform, it was inhibiting the NLRP1 when the NLRP1, it is not carrying any variants or mutation. So our hypothesis, it is that maybe the CARD8, it is really make this just equilibrium between the NLRP3 inhibition and NLRP1 inhibition, and that these three receptors, depending on the cell types, can respond in different way, or depending on the genetic variant that the host is carrying. So it is just confusing now, but we are attempting to solve this point in the next future. So thank you for the attention. And I open for question, for curiosity here.
Okay, thank you very much, Alessandra. So,
very good, excellent.
Very good. If any students have any question, we always give preference to questions from the students. You can raise your hand.
So Karine Anderson left the messages for you in the chat, congratulating for the great work, but they need both to leave. They're probably gonna get back to you later. Any students from Rio? Otherwise, I will ask a just a more philosophical question, just to warm up the audience.
No
Can I?
students yet, but I can ask after you, Gustavo.
Oh, no. Go ahead then. No, students first.
No,
Go
yeah,
ahead.
no students yet. I think we can start and maybe they will get warmed up. I can ask my question. I have a question,
Oh,
but I
you
can
have
ask.
no. Go, ladies first, Miriam. Go ahead.
That's something. Yes. So Ali, congratulations. Thank you very much for your talk. It was really beautiful to see
Thank
all
you.
the data put together. The question that I have is actually very small, and it just reminded me your data on CD8 T cells. The fact that when you activate the cells with anti-CD3 and anti-CD28, and you see a very low staining for PI. And it reminded me of a discussion from many years ago that once you activate the T cells, CD8 T cells or CD4 T cells through the TCR, they become an Extin-5 positive, and then they revert it. They don't really die. So I wonder if that staining you saw is temporarily, if these cells are actually going to die, or if you think that in case of CD8 T cells particularly, the inflammasome complex can be activated in a different for a different outcome, let's say, than you usually see in innate cells.
No, really, we think that this first PI uptake, it is okay in the without the ATP. Okay, it is just probably a temporary pause. Okay, we don't see a lot of Annexin positive staining for our experiment, but as we don't see really the LADH release, we see that really it is just the same characteristic that was you are talking about. Though this just temporary pore opening, okay, that it is temporary and not associated to cell death. Okay, so probably it is just you really need another signal, and it is interesting to think that a dump signal that was just the ATP, that is just a very, very dangerous signal, because it is just saying to the cells that the neighboring cell is dying. Okay, so it is very important normally in the physiology of the inflammation. This is important for the signal of the pyroptosis. So I think that I don't know if I just asked to your question, but really also Gustavo once told us this question of this temporary pore formation in the activated T cells, and I think that is the same point, but it is probably reversible pore.
No, that's really interesting. You did answer. I still have this hanging question on my head if all these happen, because for the Annexin-5, I always thought there was a shortage of ATP that would make the enzyme not work. But since you also have this temporary pore formation, it seems there will be something evolutionarily selected. Might be bigger than at least I was envisioning. So, but it's really interesting to see it put that way. Thank you.
Yes, we know that we see the PI uptake, and some of these dying, such as the live and dead dying, that are entering. Okay, so but it is just we don't know really what is happening there. Okay, but we know that really we don't have this great release of big molecules from the inside of the cells.
Okay, thank you.
So my question was, is there any student appear? Can I just go, because my point is a bit similar to Miriam's. So first of all, yes, I mean, it's well known that activation of cells, particularly T cells, causes a flickering of membrane, which has two main results. One is opening of small pores that make the PI comes in. And the other thing is, in some cases, even the externalization of phosphatidylserine occurs in a reversible manner, okay? But my philosophical question would be, what do you think these innate immune receptors, particularly inflammasomes, you know, has to do with CD8 biology? Okay.
Yes.
Because
Okay.
it's you know, it still puzzles me a bit.
Yes. Okay, yes, yeah, really, this is also one of our big question, okay, as since if we are just thinking about the antiviral role, it is intriguing because we are trying to think about a virus that is targeting specifically the CD8, but normally the CD8, just the NK cells are resistant to virus, okay? So this is one thing that really, if we are thinking in that just for these pathways, really, I don't know. I don't know. But one thing that we are thinking is that maybe they are just sensor for cell stress, and the CD8 cells, they are releasing a lot of, for example, vesicles, okay, because they are not just cytotoxic, they can also have secreting pathways, okay, for the cytokines. And maybe this question of inflammasome may have a role more in sensing these cell perturbance of this physiologic role of the secreting pathways of the CD8 cells to regulate, because the cells, but really we are just thinking about and open to suggestion, okay, for people that are more working with, have more experience with CD8 cells, because really it is hard to think about.
Yeah, one thing about CD8s are they are really tightly regulated to avoid any, you know, damage.
Yes.
Without context. So, but I still have a bit of it is puzzling, okay? And I'm thinking always, is there any other thing that are happening along with the system that primes or you know, make the results come as it is? One thing is that do you see any difference between naive CD8 cells and activated CD8 cells?
Sorry, it is cut your question. May you repeat this last part?
So do you see any difference when you study the influence of these inflammasome activators in naive CD8 T cells versus activated CD8 T cells?
Okay, we don't. Okay, we just do this experiment for the B cells. So I don't now. I don't remember if we have just make this. We can look at this, because this is one of the question of also that some people do at the Congress last year. Okay, about the subpopulation. I don't know. But for example, for the B cells, we have no difference in the response. So really, I don't know. I don't know what expected in CD8 cells. But it is for us. It is also intriguing that CD4 T cells, it is different from the CD8 in terms of inflammasome response. Because really, if you think that they are just innate sensor left there, we are expected to have the same behavior, but it is not. So probably they have a different role in CD8, okay? In the first paper of Kemper about the NLRP3 in CD8, they are just trying to linking the metabolic pathways of the CD8 to the activation of the NLRP3 inflammasome. So again, I don't know if it could be a question if the CD4 T cells are different from the CD8 T cells from this point of view. So maybe could be a link but really now we don't know.
No, thank you very much. I mean, we are setting up a system. I mean, already settled, Michel. These are part of the results that he was supposed to send me. I mentioned in the beginning. So Michel and Barbara is established this differentiation protocol of CD8s to TC1, TC2, 9, 22, 17. So we can approach that if it's not too much in vitro and mouse for you, because our system is mouse. Okay, but if it's inflammasome, is a general important phenomenon in CD8 cells. Why should be only human, not mouse? So I think we can try to put some things together.
Okay. Yes, I think that, okay, we don't know if we don't try, okay? So, but we know that the structures of the NLRP1 are different. So in some points, they are similar for the activation, human and mouse and rodents, okay? But not in all the points, and mouse have not the CARD8 receptor. So I think that maybe some similarity maybe exists, but there are some other type of regulation that are not, it is not possible to study in rodents. Maybe that's CARD8 or type of animal, not in rodents.
All right. Congratulations, Ale. Super.
Thank you.
Thank you.
I have a question from Bruno.
Hi, Diego. Thank you. Hi, Alessandra. It was a very, very nice talk. I was very intrigued with the different regulation for the same components in different cell types, and particularly with the BTK role. I don't know if it's just my lack of knowledge, but is caspase 1 direct target of BTK, and how the same kinase could have such different effects on different cell types, and on also on this line, are there different caspase 1 substrates that in the different cell types that may also explain these effects beyond the IL-1 beta family proteins?
Yes, Bruno, thank you for the question. Yes, a lot of these are also our question. Okay, so the first point is that, okay, the BTK apparently can affect the phosphorylation on some sites, specific sites on the NLRP3. As I mentioned, it is not sufficient to the activation. Normally, it is just an amplifier of the NLRP3. And okay, we really, it is, I think it is, we don't know how in different cells these kinase, this phosphorylation have different effect. I think that Vinicius, the postdoc of the lab, it is studying this point, okay? So we just don't know. But the point is that the NLRP3, it is a molecule that have a lot of post-transductional modification. And all of really the regulation of the activation is the sum of different signals, okay? So one explanation is that probably in neutrophils, you have other breaks on the NLRP3, and so the effect it is different. But really, it is just ongoing question, and I don't know. I don't know what is really happening. The other point is that about the substrates of caspase-1. Yes, I think that it is a very old question. Okay, so just 15 years ago, the community of Inflammasome is questioning about what are the substrates of the caspase-1 cleavage. And we know that we have a lot of substrates that are not just IL-1 beta or IL-18 or Gasdermin D. We have enzymes, glycolytic enzymes, and other types of protein that have physiologic role in a cell. So probably also this point, it is interesting thinking of different cell types and the outcomes of this inflammasome activation. But really, I think that it was questioned a lot of years ago, and then I think that people maybe don't look at this question. So for this reason, we don't know yet if we have different substrates in the lymphocytes, for example. In neutrophil, we know that we have not just caspase-1 for cleavage, the IL-1 beta, but it is another story. Okay, so we have just another molecule that are making the inflammasome working.
Sure, thank you very much. We actually have another question here.
Yes.
There's actually a comment after the discussion with Gustavo, that maybe a big difference might be between T cells and B cells and CD8s and CD4s because of the place where they react, right? The CD8 T cells are expected mostly to go to the place where the infection is happening. So they might, that difference that Gustavo asked between the naive versus the activated CD8 T cell, and depending which they are central CD8 T cell or peripheral CD8 T cell, might make a big difference with the response.
Regulation.
While the B cell might stay topologically most on the same place, it might not change much between the profiles. So it will be, I think, really interesting to look at those different populations that Gustavo mentioned they can generate, if it will be related to the mouse model in some sense.
Yes.
Because they might have, right, a exposition that will be different depending on the state of activation, if it's in the lymph node or if it's in the tissue.
Okay. This is a good suggestion. Thank you. Yes.
Can I also ask a question?
Yes.
So since you're investigating the role of these inflammasomes in all of the adaptive immune cells, and you also showed some results on B cells, mine is actually not specifically related to the data presented, but do the activation of inflammasomes change how B cells will switch antibodies, or how they will differentiate into memory cells or plasma cells?
Yes. Okay, so yes, I think that this is a point, because in general, we know that when you have a lot of inflammation, this is not good for the switch. Because normally when you have an excessive inflammation, you have just IgM production. And this data, it is, I think that is going together with this first paper that a group, just not us, published about this question, that when you have a lot of NLRP3 activation, you have a lot of IgM response, okay? And so could not be good for the IgG, for example, switch. Okay, so this is a point. How this happened within the B cells, we don't know. But I think that maybe it is related to what Bruno is talking about, this question of other types of substrated of caspase-1. So maybe you have within the B cells different substrate that can modulate it. This is the first point. And the other is the question that, okay, these sensors may act as transcription factors too. So you know that, for example, AIM2, can work activating the production of chemokines in the nucleus, and NLRP3, it is working as a transcription factor for Th2 differentiation. So maybe it is we don't know if we can have also this type of effect, that in that case it was caspase 1 independent. But I think that it is interesting because when I mention about these studies of genetics and diseases, it is very intriguing when you are now thinking about the effect of inflammasome variants, looking at the B cells and the antibody, because the studies are not so recent, and they show that some variants are associated with the increased titer of autoantibodies, for example, in some autoimmunity or Crohn's disease or whatever. So the point is, okay, we just think about that we have a lot of inflammation, a lot of autoreactive T cells activation, and it is just a consequence. But I think that now we can also hypothesize another story for this data. This is the point. So I think that, okay, we just see the point of an iceberg of all this question of inflammasome in the adaptive cells.
Thank you. Do we have any more questions? I guess we're done. So thank you very much, Alessandra, for sharing your data with us. It was very interesting to know more about your work and on genetics of inflammasome activation. Thank you very much. Thank you very much, guys.
Thank you for
Thank
the
you.
invitation.
It was really great.
The questions and suggestion of all.
Excellent, Ale, excellent.
Obrigada.
Grazie
Ciao.
mille.
Bellissimo.
Grazie.
Bellissimo.